Hi, good morning, everyone. Great to see you here, and welcome to our Wrongful Conviction Day live stream. My name is Meredith Kennedy. I'm the director of the Innocence Network Support Unit at the Innocence Project, and we're so thrilled that you've joined us today. I'm really excited for a great day of powerful programming featuring advocates, experts, and directly impacted people. This year, we're marking the 9th annual Wrongful Conviction Day. Wrongful Conviction Day is a day of collective action where the innocence community works to raise public awareness and deepen public education around the issue of wrongful convictions. The theme of Wrongful Conviction Day this year is collective impact. And with this theme, we're exploring concepts of interconnectedness and interdependence by examining the ways that injustice is experienced, fought, and repaired in community. And those ideas will be woven throughout the programming we'll bring to you on the live stream today. So just a note about the format: a different organization, some that are part of the Innocence Network and some that are not, have taken over each hour of today's program. Some segments will last the full hour. In fact, most should last the full hour, but some, particularly in the afternoon, will have some breaks built in. We are streaming the programming today via Crowdcast, but you can also tune in on the Innocence Network's Facebook page and our YouTube channel. If you need or prefer closed captioning, we recommend watching on Facebook or YouTube. We hope that you'll share what you're learning on social media today using the hashtag Wrongful Conviction Day and tagging the Innocence Network. And our handle is at Innocence Network on social platforms. All the segments are being recorded and will be available later today in case you missed something or you want to watch it again later. I really want to thank all of our live stream segment organizers, speakers, and all of you for tuning in today. And I particularly want to thank this year's Wrongful Conviction Day Steering Committee and my colleague in the Network Support Unit, Kate Murray, who was the primary architect of this incredible lineup of programming. We really hope you enjoy it. And now we're going to begin the first segment. And I'm going to turn it over to my colleague from the Idaho Innocence Project, Professor Greg Hempikian.
Hi, I'm Dr. Greg Hempikian. I'm speaking to you from my lab at Boise State University. The lab's behind that wall. We work on COVID and HIV and also forensic justice. I have a degree in molecular biology, started out in fruit flies, worked in marsupials, human skin for a year, lots of different projects. The nice thing about being in biology is that the same tools that allow me to work on fruit flies, like some of the characters behind me there in the photographs, or human beings, are exactly the same. And most of the advanced techniques were perfected in those animal systems. So today, I just want to give you a little tour of DNA through PowerPoint and talk about how we use DNA to free people and how DNA has been used in some cases to wrongfully convict. What kind of mistakes can happen? Because we always want to keep our critical thinking sharp. We are all humans, we all make mistakes, we all have very limited knowledge. I'm going to share some of my limited knowledge with you that I've gained over 20 years working on wrongful conviction cases. More than 3 dozen people we've been able to free from wrongful incarceration. Us and a whole giant team of people. It's never one person who does this. So I'm going to share some slides with you. I'm so sorry we can't interact. That's my favorite part of teaching, but we're going to have to do what we can do. If you want to get in touch with me, the last slide has ways to do that. Here's the title of the talk. That's who I am. There's an ad for a TED Talk on there. But I think you're going to get a lot out of this talk. This is a image that was presented to me by the first prisoner that we tested for the Georgia Innocence Project, where I was on the initial board. I still do a lot of work with them. They're an incredible organization. And this client told us he was innocent, and when we checked the DNA, it turned out that it confirmed guilt. And the reason I show this is because I firmly believe you cannot tell who is lying to you. Why do I believe that? I believe that because people can play poker, and the best poker players are not police officers or pastors or anybody who might deal with human character. They're poker players. Lying, deception is part of who we are. We do it to be polite. We do it to get things we want. So I I I don't think it's a good thing to do to look at wrongful conviction cases and always say how obvious they were, because I think people were fooled for a reason. It's not usually a bunch of chains of bad actors only. Juries try their best. I know jurors work hard. I've been on juries, and I want us to have a critical eye as we look at everything, not an eye thinking things are obvious. So, what is science? Well, science can't answer everything. I can't answer if this wonderful sentiment in this drawing is true or not by science. That doesn't mean it's not true. It may be true. Um, and truth is something, we think we can tell about people in the world, but we in the lab use a specific method of finding truth. You might believe, for example, you're a good judge of character, but science helps us assess that. And from the research, I know that even trained detectives are no better at detecting deception than normal people. Okay. Here's a typical kind of case that you see, I think, on a lot of the television shows and things about how the innocence organizations work. This is another case I worked with Georgia. They did phenomenal work on here. Here's a lineup. Okay. Can you tell who's guilty? So in a lineup, you have 5 people, only one of whom is a suspect, and that's the case here. Looking at their faces, can you tell who's guilty? There's all these television shows that tell you people had an instinct, or there's a way to read body language, or all this stuff. I believe most of that is garbage, because people act in very odd ways when they're in odd circumstances. I talk about the Twin Towers. What was the normal reaction to the plane crashing into the Twin Towers? Was normal the people who jumped out the window holding hands? Was normal the people who rushed into the flaming building to try to help people? Was normal people who rushed out of the flaming building running over people? I think we have to be very careful what we call science. Science means that there has been studies and numbers. So here's a lineup. We asked someone, in this case, a 74-year-old rape victim who had not been sexually active, was attacked, and she was asked to identify who the perp might have been. There was one person who's a suspect. And the person convicted of this crime wrote to the Georgia Innocence Project. Who am I talking about? I'm talking about John White, the 19-year-old gentleman in the middle there. Why is he in the middle? I'm told they asked him where he wanted to stand. He's like, right here. I wanted to get a good look at me. You see him standing there. Now I ask you, is he guilty? By the look on his face, by the way he's standing. Well, the curious thing about this is John White did not match the semen that was finally recovered from the underwear. Pretty standard story that you might hear through the Innocence Project. He was eventually freed, but the student who worked on this, when the district attorney announced a new arrest, someone who matched the evidence, who was in the database, the DNA database, when that name was announced, the law student who worked this case said, I recognize that name, James Edward Parham. And we said, well, a lot of people write to the project. Maybe he wrote, we saw a letter here, and he imagined in your head, you make those connections. No, 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 it had something to do with this case. And in fact, James Edward Parham was in the lineup. So I use this slide to remind myself that honest, sincere people like this victim make mistakes, and DNA can help us correct those mistakes. And we now know that the evidence matched Mr. Parham in this case. Okay. So what is this method of science? How can we use science to counteract bias? Now, science can also be used to enforce bias because it's all how you use an instrument. But science is really effective at eliminating bias when it's used correctly, because that's one of its main goals. So how is it different than other forms of learning? Well, for this, I'd like to quote one of my favorite scientists, Galileo, who really laid down a lot of the precepts that I try to meditate on every day as I do my work. One of them is, by denying scientific principles, one may maintain any paradox. Paradoxes are okay. Paradoxes are two things we believe that are in conflict with one another. Where are they okay? Well, they're okay in religion. They're okay in beliefs about a loved one who, continually misbehaves, but we believe in them. We maintain paradoxes, but in science, you really can't do that. And so science is restricted to things that can be resolved without having conflicts like that. In Question of Science, if you remember nothing else from the talk, remember this: the authority of 1,000 is not worth the humble reasoning of an individual. So I want you to concentrate on this, your prefrontal cortex, the reasoning part of you. I know so much of what we do in the Innocence movement is about the heart. And believe me, I have a heart. And I hurt for the people I love who are wrongfully incarcerated. But reasoning is what gets people out in my work, and reasoning helps us avoid bias. Count what is countable, measure what is measurable. What is not measurable, make measurable. Find a way to measure it. A smear of spit used to just be a smear of spit until people learned how to measure things in it, the proteins, and then the DNA eventually. And that science proceeds more by what it is learned to ignore than by what it takes into account. So what does that mean? Well, think of the Salem witch trials. And in fact, let's use them as an example, my favorite example. What the witch trials really tell us is something very essential about error that is often missed. And I'm going to teach it to you right now. When I was first starting in forensic science, as opposed to the molecular biology I was doing before, I was shocked that the field really didn't do the kind of error measurement that most of the other fields of science I was involved with were doing it. So I tried to come up with this explanation for people, why it's important to measure our error. If we measure the justice system by the number of truly guilty people convicted, the successful cases, did we get the person who did the crime? Then the Salem witch trials were an astounding success, because more than 100% of the witches were convicted. So what we need to look at, what the Salem witch trials teaches us, among many other things, is that we have to determine error. We can't just convince ourselves that being right some of the times we can measure, means that we're always right. We have to find where we're wrong. Where does our system fail? And that's the job of science. So I began to study those measurable outcomes with some wonderful people at the Innocence Project in New York, Emily and Olga, who actually did the heavy lifting. I thought of the project and did some of the work. They did amazing work on this case. And we reviewed 194, the first 194 exonerations. What kind of evidence? How many of them were cigarette butts? Were any of them, condoms? What about hair? What type of DNA analysis? And the reason we did this is to present to judges who would ask things like, well, why do you want to test that DNA? This guy's, in prison for 20 years. Nobody's ever been freed from a cigarette butt. And we'd be like, yeah, this percentage of people have been freed. So applying numbers to situations that don't have numbers. Okay, but science is also subjective, and that's where we get into some mistakes that get made, because human subjectivity, our judgment, can be off. We have bias, and so where we deviate from the strict rules of science, we can make errors. And where can that happen? Well, in visual tests. If we're testing for semen, we're testing for blood, those are color tests, like a pregnancy test or a COVID home test. They're colors, little bands. That's subjective. And some labs don't even take pictures of the results. So folks are convicted without any record of what the result of the test was. It's just the record is the testimony, the writing of an expert, but experts can be wrong. I've been wrong. So basics of DNA, just to run you through quickly. If you don't want any science, just fast forward for this. That's the nice part of it not being live. Here's a cell. Within the cell, you see the purple nucleus there. That's where most of the DNA is. There's a little DNA in the mitochondria. We sometimes use that, but nowadays, pretty much all the DNA in forensics is from the nucleus. The way we get DNA is a swab. It's kind of like a brush you can put in your mouth. Many of you have used them for 23andMe or for tests that you use, but it's out of the mouth. And you get these cells that look kind of like cornflakes, I think. These are epithelial cells. They tear open easily, and they let their DNA out of the nucleus with a little bit of detergent and some enzymes to kill the things that would destroy DNA. If they're really the same enzymes you have in meat tenderizer, the protein breaking enzymes. This is what the result looks like, and I'm not going to give you a lot on this. I'll just say here's two DNA peaks. How high the DNA peak is tells us how much DNA was copied. We copied the DNA many times to make it visual, to make it measurable. And here's one location. We're looking at the chromosomes, and the person has a size 16 and 17. This chromosome 3 location that we call D3. And the person has two peaks. The length of the peaks, the length of the DNA fragment we're measuring is these numbers in the boxes. They get bigger at one location as we move to the right. But I just want to show you what a typical fingerprint, DNA fingerprint, looks like, and how easy it is to look at, because if this matches the evidence, this is from a person, if it matches the evidence, we call it a match. And it's just these small numbers in these boxes. That's all we look at. You look at 26 of those numbers for someone, and you have them to the exclusion of all other people in the world. So that's one location. This person got a size 16 from one parent, a size 17 from another. The next locations over here, they got a size 15 from both parents. You don't have to know a lot about this. I just wanted to show you what they look like. How do we get to court or get someone back into court if they're in prison? Well, there are 10 steps to profile. First one is you have to identify the stain, or you make an attempt to, if there is a stain. You can do that with an alternate light source, like a black light that makes things glow. I'll show you. Certain things glow pretty bright under black light. That's not a confirmatory test, but it shows you where you might want to swab to pick up some DNA. And then we have to move the DNA to water. That's what those swabs are for. You rub it, they're like Q-tips, and then you put it into a little tube. The tube is very small. It's no taller than a quarter standing on edge. Some of them as tall as a dime standing on edge. Very small test tubes. Or we can also use things like a wet dry vac to get DNA out of clothing with this MVAC as the device that most people use. We have to get the DNA out of the cells. We break open the cells with some detergent and remove the debris and get the DNA into the water, and we suck the water out. And then we have to tell how much there is, how much DNA. We can tell how much male, how much total DNA, therefore how much female DNA, and how degraded it is, how broken up it is. We copy what's there, the pieces that we want to copy in size. We copy them 1 billion times, hundreds of millions to 1 billion times. These chemical kits takes about 90 minutes, and that makes the DNA visible to us. Beautiful invention called PCR that won a Nobel Prize. Polymerase chain reaction. You can watch it on YouTube. And then we run it through copper straw. The little pieces go faster than the big pieces, and that's how we measure the sizes of DNA. And then we have to interpret them, do statistics. Maybe we use computer programs for that. And then reports, and the wording of the reports is very important, and you get a bias in the way reports are written. There's a strong law enforcement bias, for example, I believe, because they don't say when lab scientists disagree. That doesn't appear in the report. You just get the higher up's opinion. You don't get anything about any discussion that might have been done. Hopefully that's changing. And then there's testimony in court, and that's really where it counts. It's what the triers of fact, the jury or judge, find to be true. That is the effective truth. That's what the result is. Okay. Have I seen errors at different steps, and how can they be prevented? Let's take a look at some of the steps. Here is that first step, looking at evidence with an alternate light source, a black light. You've been in clubs or Halloween decoration, ever seen the black light, and some things glow, and one of the things that glow is semen, so we might check underwear with a black light like this to see where are there stains. Can we test them? And then we do a chemical test to see what body fluid it might be. And I'll just show you quickly. They're like the COVID home test if you've taken one of those. Here's the line at the top just means that the liquid got to the top. You put the stained liquid, a drop from the stain in here. You put water on the stain, and you put a dropper in here, and it goes underneath because plastic here, just like the COVID test. If it gets to the top, it just means the water got up that far, it wicked that far. This is the test line. Here's a positive control test. That means we know there was semen here. Here's a negative. We know there wasn't semen there. The lab does this control. Here are 2 findings from an actual case that I worked on. And what do you see? Do you see that line that says that it tested positive for semen? Well, not so much. But this one, the analyst saw a little something, and I believe the analyst saw a little something. These tests sometimes show you, little shadow bands, and said it was a weak positive result. What does that mean? Well, first of all, many labs don't give us these photos. They just call it positive, and then throughout trial, it's called the semen stain, even though it's questionable whether it was semen. How does that make a difference? If you're the father, the boyfriend, the stepfather who's accused of molesting a child, the clothing of that child might be tested, and your DNA is probably going to be found on the clothing, especially if you do the laundry, which in this case, there's a documentation that the man did the laundry. And that's normal to have your DNA on your child's, intimate clothing, maybe even on their body. Having something called semen that also shows with your DNA profile, that's a different story. And so by calling this stain a semen stain throughout the trial, I believe, in my view, a false impression was conveyed to the jury. Here's more testing. Here's a positive control, different kind of test, an acid phosphate test for semen. There's the positive. This is a negative. And you can see a little pink even in the negative. This is just using laboratory, known semen, known water, and you still get a little bit of pink. And here you get two tests from the actual evidence, and they're seeing a little bit of pink, but does it look like this? Is that a positive? Is it a weak positive? What does that mean? Should we call it semen through the trial? These are the kinds of things that are subjective, and even though they're sciencey, they're subjective. There's no number here. There's no measurement. Galileo would be very unhappy. There are machines that can tell you how red or pink something is, give you a number, but they didn't do that here. So the question is this positive or negative? It's a subjective call. Do we see positive tests or in any of these, or is that just one line? Well, the lab in this case said one of them was a weak positive. Okay. Something else, just using common sense. What other mistakes can happen? Well, the order of operations is critical. This is from one of our restrooms here at the university. And there's a restaurant there, and they tell the people who work in a restaurant, make sure you wash your hands after you use the bathroom. Before, if you don't wash your hands, maybe the cook comes in and puts some, grease on the flush handle or something. Well, that's annoying, but it's not going to cause disease. However, if they leave the bathroom with feces on their nails, under the nails, and that's why it says get under the nails, then they're going to cause disease. And so when you wash your hands is important. The same thing with evidence in the lab. I try to get the labs to process all of the evidence first before they bring in an accused person's DNA sample from their mouth, right? The reference sample. Because we're making billions of copies of these DNA molecules, and we can spill a few. So do the evidence first, then do the sample, the reference sample. Well, most labs were doing that, 10 years ago. Now I see a lot of labs that aren't for some reason. I don't understand why they've stopped doing that. And why is it important? Well, this is how small everything really is. These are those little tubes I'm talking about. This is in my lab. You can have up to 96 of them on something like this. Sometimes we put the liquid right into holes on a plate that looks very much like this, and it's left open. And you could mix a few molecules, as you're doing this. It could be on your glove, right? It can be on the instrument. It could be in the air as you're pipetting, as you're putting liquid from one place to another. So any DNA that's transferred before our copying process will look just like real DNA. The copies are you can't differentiate between the copies and the original. It's not like a regular fingerprint. You give the police your fingerprint, they can't plant it everywhere. It's pretty hard to do. Okay, so I just try to tell labs, keep the suspect's DNA away from the evidence. Process all the evidence first, then the suspect's DNA. But what do I see? Well, here's a sheet that shows me what was in that little plate with the 96 wells in it. And look, they have the evidence right next to the suspect sample. Is there a possibility of contamination? Yes. Is there any reason to put them next to each other? No. You can run the reference samples next week. Nobody's going to court that fast. And the good labs do. So am I just being fussy? No, scientists make mistakes. This is a case where the Las Vegas police apologized to a man who had been in jail, I think, 4 years. And what happened is two suspects were accused of something, and the lab mixed up the two tubes. They sent the wrong guy to prison because somebody mixed up the tubes. And that's why putting in practical, common sense things like the order of operations might prevent this. And how do they find this out? Well, the California lab called the Las Vegas lab and said, we have a DNA match to that evidence you loaded up. And they're like, no, we already have that guy in prison. They're like, no, you don't. He's here in California. Yes, we do. No, we don't. Yes. Everybody retested. They did. And that's why Las Vegas had to apologize. They let him out. They did apologize. Am I being fussy about the contamination? Can things go? Well, this is Carlton Gary. This case was being worked by a lawyer in Georgia that I knew. He called me to be on it. Carlton said he was innocent. He wasn't really the stalking strangler that had been accused and convicted in a couple of the cases of raping and strangling older women. And we finally got DNA from one of the cases, didn't match him like the first time DNA was gotten from these cases, and he's excluded. There is someone else's semen on one of the women who had been raped and killed, who was not sexually active until her assault. And we thought Carlton was going home. And then I got the most unusual letter I've ever gotten. Well, first of all, they got a hit to a modern crime in Georgia, a gun crime, and we thought they found the guy who left the semen. But the guy was too young to have left the semen. This was a 30-year-old case. The guy was in his 20s. But he had a jacket that the gun was in. So we thought maybe the jacket was an old Army jacket. Maybe the owner of that jacket left the DNA. Anyway, then I got this letter, the GBI, Georgia Bureau of Investigation, found out that the sample from those two independent cases that was loaded up all the way to the FBI national database was a semen sample prepared by someone in the lab who worked in the lab, or a husband, I'm not sure. Someone who worked in the lab brought in a semen sample to test equipment in the lab years and years ago, and somehow it contaminated two modern cases. One a very old case that we were having tested, and the other a gun case, not being done by the same analyst, not being done with the same instruments. Nobody knows how this happened. So it is possible, as a little sperm cell, it is possible that, and proven, that DNA does last, that even though we try to wash it out with bleach, etc., there is contamination that happens in the labs. And we have to be aware. Does this mean DNA is bad evidence? No. But I deal with a lot of cases, or oversee a lot of cases. There are, these labs are doing thousands of cases. How many errors are being made? Some errors are being made. All right, what about cognitive bias? What about story? Well, I worked with Idio Dror, who's a cognitive neuropsychologist. He looks at bias, and he published this paper. This is not my work. He worked from a study that a pathologist had done. These pathologists are medical doctors. They testify. They decide whether a death is accidental or homicide. The most basic decision that starts the ball rolling in a homicide investigation. And what Idio did was he gave the exact same medical evidence to more than 100 pathologists. I can't remember it was, divided them into two classes. One half got one story, and another got another story. Same medical evidence. And the story was, in one case, the white bar here is that it was a white child in the care of the maternal grandmother, the mother's mother. And the black is, it was a black child in the care of the mother's boyfriend. And in that case, the story of the white child with the maternal grandmother, the ones who found, some of them said it can't be determined, but the ones who found whether it was homicide or accident, some 28% or so said the grandmother of the white child, it was just an accident, no homicide. The accident for the boyfriend of the black child was, only about 7%. When you look at the determinations of homicide, these are people who are going to be investigated for homicide, right? Then the story of the caretaker, that was the mother's boyfriend, black child, goes up beyond 30%, say it was a homicide. That means this is going to be a murder investigation. And the really interesting part of this is published in a really good journal, Journal of Forensic Sciences, and they have never gotten more calls for retraction. The pathologist really got upset. Others got upset, saying, it's okay to consider who the caretaker was because we know that unrelated people are more likely to commit murders. I can't remember. I don't buy that, and I'm dismissing it. But there are people who do think, oh, it's okay for these medical examiners to consider things about the population in general and then infer about the specific. That is, a specific person in this case may have committed homicide. In fact, they're going to say it was definitely a homicide. Maybe this person committed it, based on the fact that a boyfriend was taking care of a black child versus a grandmother was taking care of a white child. If you have a problem with that, and this was just published last year, and he has a new article this year. I encourage you to look up Idil Jor's articles. Well, can this happen in DNA? Can there be cognitive bias? Can you tell someone a story, as scientists like me, a story, and us become biased? Yes, we're humans first, scientists later. You know, you're a scientist 8 hours a day. You're a human even while you sleep. And so we had a case, I had a case from Georgia that Rodney Zell, a private attorney, had taken. A man claimed he was innocent, and Rodney got in touch with me in the early 2000s. And we started working this case together to investigate if this man was innocent. It was a multiple perpetrator rape case. 3 men raped a woman. There was DNA and a mixture of semen, mixture from these men that clearly there were multiple contributors in the semen. And Kerry Robinson, the analyst from the Georgia Byrd investigation, said he could not be excluded from this evidence. Now, because of that, the man who admitted to the crime, to being one of the 3, to being the ringleader, having shot someone and raped this woman, they offered that guy a deal. And because the DNA analyst said, "Oh, Kerry's not excluded," and they asked this guy who did it. The reason they looked at Kerry was he said, "Oh, the guy I know was with me, Kerry," and he gave a different last name first. And then he was like, "Oh no, it was Kerry Robinson." And Kerry said, "Oh, he's just saying that because he thinks I ratted him out to the police." So when it's one person's DNA, it's a clear answer usually for us, whether it's a match to the evidence or not. But when you mix together several people, it becomes very challenging. And the metaphor I gave is this. Sorry, I was trying to explain it to a prosecutor at one point. I took the letters from my name and his name. I said, well, if we take the letters from your name, just you, letters from my name, just me, and we put them into a bowl with Scrabble tiles, can we pull out more than just the 2 of us? That's a mixture, and mixtures will give us an interpretation that cannot exclude lots and lots of people, but not all of them contributed. And so here is the analogy. I gave this to our poet laureate, Diane Raptosh in Idaho, and I gave her about 10 minutes. I said, can you see names in here besides my name and this prosecutor? And she found these. I think I gave a couple of them. So I like, Oprah, Ellen, Magellan, Gilligan, Ginger, Marianne, and Mr. Howell from Gilligan's Island. So mixtures, when it's unclear, people can see things. Scientists can see things. And I think that's what happened in Kerry Robinson's case. And I tried to convince a judge of that. We did a study, Idil Jor and I. We took the evidence from Kerry Robinson's case and we gave it to 17 examiners at a crime lab outside of Georgia during one of the trainings. We didn't let them talk to each other. They had, because when scientists talk, they're just like everybody else. They come to an agreement. We said, hey, these are scientists. They're trained. They're forensics examiners. They know what they're doing. Just come to your own conclusion. Out of the 17 analysts, only one agreed with the GBI. And 12 examiners excluded him. The rest found it was inconclusive. So we brought this to the judge. He didn't care. This case went nowhere. I tried everything I could. We did a show with Fox News Atlanta. That was great. We swabbed people at the news station and found out they had more in common with the rape evidence than Kerry Robinson did. A 22-year-old white intern woman had more in common with the semen evidence than Kerry Robinson did. Didn't matter. The judge didn't buy it. We got press on this. The Economist came up in a New Scientist magazine. Still, nothing happened. Finally, I asked the lawyer to write the GBI, Georgia Bureau of Investigation, as we were doing all this, and say, how much evidence? What do you need to exclude somebody? And he said, George Herron said, even if there's no DNA in common from a suspect in the evidence, we can't say they're excluded necessarily. And so Georgia was using the term cannot exclude, which means a match to most labs. When we say we cannot exclude, we give a statistic that means a match. They were using it as, can't tell. That's very different. Inconclusive is not cannot exclude in my field. And I found this abhorrent. I understand what he's saying here. I understand the logic of it, but it is not the way the term was being used in the field. It is not the way the analyst from the GBI testified, making it sound like Kerry Robinson probably contributed. That was wrong. This inspired a New York Times article. I won't go through it, that I wrote an essay that talked about a study the government did that showed that 74 out of 108 North American crime labs, given contextual information about a crime, a bank robbery in this case, if you give people the information, the scientists, 74 out of 108 included somebody who was not a contributor to a mixture. And the authors of that study probably have some things to debate with me about this, but I stand by it. That was a breathtaking result. That 74 out of 108 labs tested by the National Institute of Technology, Standards and Technology, got it wrong. And because they were given evidence, they were given a story. Okay, so we started using, I heard about this new computer method that eliminates some human bias, and probabilistic genotyping, it's called, and it could interpret mixtures where people have been falsely included. And the first people we got out were two men from Indiana working with Fran Watson there at her law clinic at Indiana University, and Mark Perlin of a company called Cybergenetics that made this program. And we were able to free Daryl Pinkins and Roosevelt Glenn, two great guys who had been falsely convicted again in a multiple perpetrator rape. So I was really hoping we could do this, for Kerry too, but the Georgia Bureau investigation said they couldn't find the DNA records in the computer for us to analyze. It took us more than 12 years to get those records. They found them. Can you imagine just waiting to get these records all those years? I can't imagine what Kerry went through. Here's another one, Johnny Lee Gates in Georgia, another case. He admitted to murder. He was convicted, sentenced to death. Eventually, they waived the death penalty. In 2016, the Georgia Innocence Project, the Southern Center, and my lab at Boise State University with our Department of Justice grant started working on post-conviction. And here were some of the issues in this case. Mental deficiency. They brought him to the crime scene for a confession, let him touch items. The prosecutor struck all the black jurors, not just in this capital case, but in all the capital cases from that county until they weren't able to anymore because more black people moved in and they only had so many strikes they could use from a jury. DNA was never tested in this case. We got it tested. It was analyzed by TrueAllele, again, Mark Perlin's program. This, by the way, was the notation of the prosecutor. There was an N besides the black jurors. There was a W in front of the white jurors. And if you look at the statistics, these were capital cases, death penalty eligible cases with black defendants from 75 to 1979 in this county. These are the cases, and these are the defendants. The qualified jurors called, the jurors struck by the prosecution, qualified black jurors called, there were black jurors called. The ones that were struck by the prosecution, oh, look at this, 4 black jurors called, 4 struck, 8 8, 4, 4, 4, 4. Finally, until we get to where they have so many black jurors, they struck 10. And so they ended up with 2 on that one jury. So bias and science. And we try to use science to counteract bias. It's not always possible. Science can be used in bad ways too. The good news was through the hard work of all those organizations and individuals and lots of others. As I said, it's always like a giant team of people who keep handing the baton one to another or work together. And he finally got out after 40 years. The DNA went from inconclusive from the normal techniques to an exclusion with probabilistic genotyping. Science counteracting bias. He spent 26 years on death row. 40 years total. And I got good news. There's a quick talk, so I won't be able to talk about it all. But the Georgia Innocence Project joined Rodney Zell and myself on this case in 2019, I think it was. And in 2020, we got the results because we finally got all the files. We were able to use the probabilistic genotyping. And the reason they freed him is because that same company, TrueAllele, supplies the software to now the Georgia Bureau of Investigation. And we asked the founder of TrueAllele, the guy who wrote the program, could you take a look at this case? Because what happens is when these labs adopt probabilistic genotyping, they adopt it for the new kits they're using now, and they don't look back in their old cases where it could be used to free people. I find that, again, unethical. But I understand it's efficient, but we have a chance to correct mistakes. And it took a lot of hard work. I was on this case, I don't know, 12, 15 years. A lot of other people were on it. Poor Kerry was on it for 17 years in prison. Okay, finally, I'm going to talk a little bit about forensic genealogy. Some of you sent your DNA away, and now you can say, I want that DNA to be used to solve murders as well. It's an opt-in program. GEDmatch is the big company now owned by a company called Verigen. These DNA databases move around from company to company. And how does it work? Well, basically, we're just going to look at how long a piece of DNA you have in common with somebody, because DNA tends to break and get shuffled when we make sperm and eggs. And so the longer the piece, the DNA, the less shuffling, the fewer shufflings there have been. And that means the closer relative you are. We're just looking at how many times, between two people, they have been shuffled through the process of making sperm and eggs. And if you're brother and sister, you got your sperm and egg from the same source, you're going to have a lot of DNA in common. A lot of your pieces are going to be just continuous. They didn't get shuffled much. So that's what this diagram is showing you. And you can then use these nifty tools on 23andMe or Family Tree or any of these providers, Ancestry. They all have these browsers. You can go to third party. You can see how big a chunk of DNA on a chromosome you have in common with someone else. That's what's in purple here, the shared regions. And from that, you can get a list of people who were related to you. Well, we started thinking back in 2012, we might be able to use this before it got so fancy. We might be able to use even crime lab data, Y chromosome crime lab test to get last names. And we proved that was true. That was done here in Idaho. I'm very proud to say this. Christopher Tapp, he was at my house last night. He was convicted of murder and rape in '98. That's him hearing that he was convicted on the left. That's him as a prisoner on the right. There was semen from the victim. The DNA didn't match Tapp. She was raped and killed. I won't go through the details of this. I'll just tell you, we started this case in 2007. We worked on it a long time, all the way through to the end. and other groups joined us during this, including at the end, the Innocence Project in New York. Judges for Justice got on with his Dateline, did a great story. I mean, this story's been everywhere. But the main thing is we worked with for this talk, we worked with the Idaho Falls Police Department here in the lab behind me to show them how they could use this novel technique to find from their Y chromosome DNA test they did on some semen, how they could get a last name. And we convinced them to use this technology. They then did the investigation, and eventually we got a last name, the lineage of the person who left the semen. Chris was freed after 20 years in prison. The marvelous story is that was 2017. We got some of the semen from the case. I sent it to a ancient DNA, a lab that studies Neanderthals, and it's still there. And then CC Moore was working with the Idaho Falls Police Department. You might know CC from her TV shows and all, and her great work in investigative genetic genealogy. And they were able to take that lineage that we got with the last name and find out that, in fact, the last man in that lineage, his mother didn't give him that last name, and that's why he was a little hard to find. So thank goodness for Cece and her team. And because of that, that genealogy match, not only did Chris get out, that's him on the right being hugged by a friend, but a new suspect was identified through that lineage. And he had the name that we had identified, Ussery, but his mom decided not to give him his father's name. And so that really was paper genealogy, people looking at obituaries and finding the names and numbers of family members didn't match at some point. So some great work all around. And he's admitted to the crime and is presently serving for it now, after Chris had already spent 20 years. The good news is Chris did get compensated. The last story I'll tell you about is Charles Fain, a 9-year-old not far from where we are here, was raped and killed, and Charles Fain was convicted. He was sentenced to death. He had been 4 hours from execution once. He did all of his time in solitaire, and he was finally exonerated in 2001 by his lawyers, Fred Hoops, and now I can't remember the other guy's name. I came to Idaho later and wanted to help the police find the real perpetrator of this. I knew they had some of the pubic hairs left from the assailant. And we tried over the years, in my lab, we did a bunch of the people they thought would match the DNA, never did, because we could do mitochondrial, this kind of DNA from hair that the crime lab wasn't doing. We helped the police. And then finally, I met Ed Green at Santa Cruz University, and he studies ancient DNA. And I had asked him, if he could take a look at this pubic hair and find, can you get like anything out of it, genealogy information? And in fact, he was able to. There's Charlie when he got out. We also worked while Ed was working on the DNA. We were working on a compensation bill. That's Robin Long, my lawyer at the project, Christopher Tapp, who was exonerated, and Charles Fain, who I'm speaking of now, and me, lobbying for or testifying for compensation bill. Some great help from the Innocence Project in New York and some local lobbyists, and it was unanimous. They wanted to compensate both of these gentlemen, so they got compensation. And then Ed Green's work on the pubic hair. I met Ed at like somebody was pitching a show to the History Channel. We were working as experts on the show, poking holes into graves of Hatfields and McCoys. And I asked Ed about this, and Ed was able to get a profile. They were able to do forensic genealogy on that. And there has been a charge now, against someone. So we'll see how that trial goes. But Charles is compensated. There's a new man, a new suspect in this, and there's a picture of my colleague and friend Ed Green, who worked with us on this case and really broke it open. So that shows you how science can work. It's a lot about friendship, meeting people, daring approaches, and trying to get past the stories that we tell, the bias that we have. And I think science can do that when it's applied correctly. It will never be perfect. There will, every medicine comes with new side effects. Every forensic technique will result in wrongful convictions, but fewer wrongful convictions is what we're after, and certainly not the same type of mistakes. So the type of DNA that was used is the type that 23andMe uses, and Ancestry, and all these, called single nucleotide polymorphism, SNPs, where you might hear next-gen sequencing, where they sequence a lot of the DNA, not just the few pieces we measure currently in most crimes. And this is the real future of DNA. It can also give you information about hair color, eye color, from a stain, skin tone from a stain, from just a biological substance. So some people claim they can draw pictures of people. I don't really believe that, but there are companies selling these pictures to police departments. And so a 62-year-old inmate who was already in prison got charged with this crime. And there's Charles Fain when he got his compensation from the Idaho legislature at 72 years old. He bought himself a nice used truck, a Chevy Colorado at 40,000 miles. He said, man, wow, is this ever nice? Charles is a wonderful guy, and hope he's getting a lot of fishing done. Thank you very much. I'm so pleased to be able to speak to you. And if you have comments or questions, you can get in touch with me. That's Calvin Johnson, a case I did not work on. Peter Neufeld at the Innocence Project in New York got Calvin out, but Calvin and I wrote his autobiography. So I recommend that book. Barry Scheck did the afterword for us. And you can get it almost free now. I think the copies that are being pulled from libraries available on the internet. We won't get any money from those copies, but they're generally good copies. And if you want to contact me, it's forensicdnaanalysis@gmail.com. I'm in Boise, Idaho. I wish I could be doing question and answer with you now, but I really appreciate your time. I hope this was educational. I hope it was enjoyable. And I really feel honored to be able to speak to you. I hope you learn a lot from the whole series. Bye-bye.